Thank you. It's really a great pleasure to be here. I've known about Norrie disease and worked on Norrie disease for 25 years or more. My wife, Sandra Brakefield, helped discover the Norrie disease gene. And then somewhat later in my laboratory, we worked on trying to understand what happens with the ear of a Norrie patient. And so today I'll tell you a little bit about what goes wrong in Norrie disease in the inner ear, what kinds of new gene therapy methods are being developed to treat this and many other kinds of disorders, and I'll tell you a little bit about one instance in which we've been able to treat deafness in a mouse model of another kind of deafness. So let me start by giving you a little bit of a tour of the inner ear. And if I were to cut a cross section through my head about this way, what you can see is that you would go see the external ear, the pinna, which collects sound. The sound is funneled down the ear canal to where it hits the eardrum. So that's the outer ear. The eardrum is connected to 3 bones of the middle ear. And so this is a moist, fluid-filled space that serves to couple the vibration of the eardrum into the inner ear. And the inner ear actually has the organs that serve 2 completely different senses. One is the sense of hearing, and one is the sense of balance. But they use the same kinds of cells, so it's not surprising that if there's a problem with hearing, there's often a problem with balance as well. So let's look a little bit in more detail at the inner ear. Here's a beautiful engraving from a 19th century anatomist. And you can see that there are actually 2 different, again, as I said, 2 different organs. So on the right side, there's the cochlea, this snail-shaped structure that gives us our sense of hearing. And if we were to look inside it, and I'll show you this, there's a ribbon of very sensitive cells that can convert the vibration of sound into an electrical signal that the brain can understand. And over on the left are the organs that give us our sense of balance. There are 5 of them, and each one of them also has a patch of these same mechanically sensitive cells that convert a mechanical stimulus, in this case head movements and so forth, into a neural signal that the brain can understand. So let's kind of dive into the cochlea, and we'll cut a cross-section through just one turn of these 3 turns. And you can see here that each turn is mostly a fluid-filled space, which is lined with cells all around it. The cells that I study are located right here, and these are the sensory cells that actually respond to vibration. But we'll talk also about these cells here that are sort of an energy source for the cochlea. So let's zoom in a little bit more and look at these cells. So these cells have a peculiar name. They're called hair cells, not because they have anything to do with human hair. They're about each cell is about 1/5 the diameter of a human hair. But because each cell has a little tuft of cilia coming out of the top surface. And there are 2 kinds. One is called an outer hair cell, and here's the top of an outer hair cell. And you can see that each outer hair cell has about 100 of these little cilia. And so the early anatomists thought the cilia looked like little hairs, and so they called them hair cells. You can see that they're kind of rows, and each of the rows increases in height past the previous row. And then there are also these inner hair cells that have a slightly different function. But again, each inner hair cell has a tuft of cilia coming out of the top surface. And let me just sort of draw that schematically. Again, you can see the inner hair cells, the outer hair cells. And these cells have a special sort of a structure that's called the tectorial membrane that sits over the top and connects to the tops of all the cilia. And when sound comes in, the tectorial membrane vibrates, moves the cilia back and forth, and that's the signal that allows these cells to generate an electrical signal. And then these nerve fibers that I've colored in green carry the signal to the brain. And again, here is in a cross-section where the hair cells are, the lateral wall that I'll talk about in a moment, and then where these nerve fibers then carry the signal to the brain. Incidentally, a cochlear implant would be placed right about here and would electrically stimulate the nerve fibers directly if the hair cells aren't functioning anymore. But let me turn now to another part of the ear, or a part of the cochlea, and this is called the lateral wall, or sometimes called the stria vascularis, and it has a really important function for the ear. So the hair cells use a lot of energy in carrying out their function, and they, in essence, need the battery recharged. And the cells up here draw energy from the blood to recharge the battery that makes the hair cells work. And in order to get that energy, they have to have a lot of blood vessels that come in to bring energy into that region. And again, the early anatomists, the 19th century anatomists, understood exactly what the blood vessels looked like in the ear. So here's the picture I showed you before, and here's another picture from the same volume where the scientist has shown the blood vessels. You can see all these blood vessels in the balance organs, but now look at the cochlea, this dense, dense meshwork of fine capillaries that are bringing energy to that lateral wall. And so we kind of wondered when we started working on Norrie disease, we knew that there was a problem with the blood vessels in the eye, that they didn't form properly. And we wondered whether there might be a problem with the blood vessels in the ear also in Norrie disease. And so we were able to get a mouse in which the Norrie disease gene had been engineered to be inactive. And then we could see what's wrong with the mouse when it doesn't have a proper Norrie gene. One way of figuring out the blood vessels is to fill them with a fluorescent dye. Let me just go back one. So here's this lateral wall, and now let's look at it from the side. And you can see here in a normal mouse, there's this wonderful bed of capillaries that kind of goes back and forth and brings blood, brings energy to the cells of the lateral wall. And those cells, as I mentioned, kind of recharge the battery for the hair cells. But now look, here's a mouse that lacks the Norrie gene, and it has some big vessels, a few big vessels, but you don't see the fine capillary branches that would bring blood into all the cells of the lateral wall. And here's another picture kind of looking from the top. Here's a normal mouse, and you see these wonderful capillaries going all the way around the cochlea. And here's a mouse lacking the Norrie gene. It has a few big blood vessels, but not the fine capillary branches that are necessary to bring the energy in. So this kind of work, and work that others have done since, have made it more clear what the Norrie disease protein, Norrin, actually does. And it's a little bit like a signpost that cells put out in order to draw the blood vessels to them. The cells make the Norrie protein, they push it out of the cells, it's right out there, and then when the blood vessels grow in, they recognize that sign and say, okay, I'll grow over here, I'll grow over here, and I'll grow over here. And when the Norrie protein isn't made, the blood vessels don't grow in. They never got that signal. And so we don't get the fine capillaries. What actually is a consequence then of not getting the capillaries growing into the lateral wall and not being able to recharge the battery of the hair cells? So it's a little hard to see, but let me describe. Here is, again, a cross-section, the lateral wall and the hair cell region in a normal mouse. And at 3 months old, which is not so old for a mouse, you see a nice dense group of cells in the lateral wall, and you can see the 4 hair cells in each section that you're supposed to see. And even at 15 months, which is pretty old for a mouse, you still have this nice network of cells in the lateral wall, and you still have the hair cells. But in a mouse that's lacking the Norrie protein, by only 3 months, you're starting to see cells die. You see these holes up here in the lateral wall? That's not good. And the hair cells are still there, but if you wait to 15 months, now many of the cells in the lateral wall have died. They've not gotten the blood vessels that they need. And because they're not supplying energy to the hair cells have also died. So this is what's happening in the ear, that slowly over time, the energy is not getting to the right cells, and the hair cells die. And when the hair cells die, then there's no sensation of sound, and that produces the hearing loss that unfortunately so many of you are familiar with. Okay, what can we do about that? And I'll tell you a little bit now about the new methods that are being developed for gene therapy. There are a great many labs around the country and around the world that are very excited about gene therapy. And in general, what they're using is different kinds of viruses, especially this one called adeno-associated virus. And the reason they're using these AAVs is that the viruses over hundreds of millions of years have gotten very good at getting into a cell and carrying DNA into the cell. So scientists have figured, okay, well, we can kind of hijack this use. Let's take a virus that's good at getting into cells, but put our own DNA into the cell in order to get the DNA into a cell where it might otherwise be difficult. It's a little bit like a Trojan horse, where the cell recognizes the virus, it pulls it inside, but in this case, the Trojan horse is filled with, not with soldiers, but with doctors, with a kind of DNA that would be able to correct a problem that is inside the cell. So here's the way that AAV, the adeno-associated virus, can be used in different ways to correct a genetic problem. And let me describe several different kinds of genetic problems that can be corrected in slightly different ways. So here's a very classic sort of genetic problem where you might have a recessive disorder. That is, the father might have one bad copy of a gene, the mother might have one bad copy of the gene. And if the child is unlucky, they would inherit both bad copies of the gene, and then that gene wouldn't make the protein properly, the protein wouldn't function, and that could create a disease. But we can use the adeno-associated virus and put a good copy of the gene inside the virus, and then the virus can carry the gene inside the cell. And so the cell might have a couple bad copies, but now we've added a good copy, and the cell can make the normal protein, and then that cell can start to function properly again. So that's a very simple sort of example. Now it happens that AAV is great, but it can't actually hold that much DNA. So it can hold a little more than 4,000 bases of DNA, but there are many genes that are much bigger than that. Many of the deafness genes that I study have DNA that will not fit in an AAV. So other people have gotten very clever about using AAV to put in the gene in two halves. So you might put half the gene in one AAV, the other half of the gene in the other AAV, and if you can get both of them into the same cell, then some sort of magic happens inside the cell. The two halves recombine, and now we've got a good copy of the gene inside the cell, even though it was too long to fit into one AAV. It's not as efficient, but at least in animal studies, it's been shown to work. Now, there are other kinds of genetic disorders where a child might inherit only one defective gene, but there's something about the defect that hurts the cells, that creates disease, even if the person has another normal copy of the gene. So this child might have inherited only one bad copy of the gene, but that gene does some damage. And so we'd really like to be able to get rid of it. There are other ways of introducing genes into the cell to get rid of a bad gene. In this case, for instance, the adeno-associated virus doesn't carry a normal copy of the defective gene. Instead, it carries DNA that can make a corrective protein. In this case, one that's been used a lot is a protein called Cas9. This is the protein that's at the core of this CRISPR method that you may have read about. And the Cas9 can specifically recognize one bad DNA molecule out of 3 billion in the genome. The Cas9 can go over and basically chop up that gene, and now the cell has no bad copy and one good copy, and so it can function properly again. There are yet other methods. There's one that's just been developed and hasn't been used in any animal models yet, where instead of supplying a corrective gene, the adeno-associated virus has the DNA for a new kind of molecule that's actually a fusion of two different molecules. One is the Cas9 that's really good at recognizing single positions of DNA, and the other is an enzyme which is called a deaminase, which actually changes the DNA to make it go back to the normal copy of the DNA. So rather than replacing the defective DNA, this new method, which was developed just in the last year in Boston, can actually correct, it can edit the DNA that's in the cells themselves. And then there are yet other methods. For instance, if the cells that have a defective gene have actually died and are non-functional, then you can't use the AAV to carry DNA into them. They're not there anymore. But you could use the AAV to carry a gene into other cells that are nearby to restore the sort of function. So for instance, if the light-sensitive cells in the eye are no longer there, you might use the AAV to confer light sensitivity by making a protein in other cells in the retina and begin to restore function. Okay, how does this apply to Norrie disease? Which of these methods might be appropriate? So as many of you know, in Norrie disease, the mother might have one normal gene and one gene with a little bit of a defect. And if a son inherits the copy with the defect and the Y chromosome from the father, then the only gene that is there is defective. But it's a fairly simple matter, using the very first method that I described, to try to use a virus to carry a normal copy of the Norrie gene into the cell. Now, it happens that the Norrin protein is really a tiny protein, and it only requires 400 bases of DNA for the instructions for how to make the Norrie protein. So this only takes 400 bases, but the AAV can hold more than 4,000 bases. So it's really easy to fit the Norrie gene into an AAV. So what we might imagine then is that the AAV, we put the gene for Norrie protein into the AAV goes into cells in the ear or the eye, and now there's a good copy of the gene and the cell can make the proper protein. So nobody is trying that at the moment, but that would be the strategy that would be most straightforward if one were thinking about gene therapy for Norrie disease. Let me tell you about some other studies that we've done where we have been able to use essentially the same strategy to correct hearing loss in a mouse model of a human disease. So let's go back to the hair cell region. And in the hair cells, we know a lot about the molecular apparatus that's used to convert sound into a nerve signal that enables us to perceive sound. And if we look, I won't go into a lot of detail, but if we look at the very top of those cilia that are on top of each hair cell, there's this complex of proteins that we've just recently begun to understand, maybe 6 or 8 different proteins in this huge complex. But one of them here, there might be 2 of them in each complex, has this rather gangly name of LHFPL5. Just a miserable name, but LHFPL5 is what the name is. And if there are patients that are missing LHFPL5, then they have congenital hearing loss, they're born completely deaf, and they have no sense of balance because the hair cells in the auditory part and the balance part of the inner ear simply aren't working. The LHFPL5 is also a small protein. It takes a little more DNA than Norrie for the instructions, but it's still easy to fit the DNA for LHFPL5 into an AAV. Here just shows you again, unfortunately this gene has 2 names, the LHFPL5 and has another name, TMHS. And so sometimes you'll see this thing labeled TMHS and sometimes LHFPL5. I'm sorry about that. But we can see here that this protein, the LHFPL5, is marked in red, and it's right at the tips of those little cilia on the top of each hair cell. As I mentioned, if it's defective, it causes a human deafness that's called DFNB7. And there is also a mouse that happens to have a mutation in the same gene, so we can study the mouse in order to understand the human deafness. This mouse is called hurry-scurry because it has some balance problems. So looking at the inner ear of a mouse with a defect in LHFPL5, here is a mouse that is only missing one copy of the gene. And I'll just show you this electron micrograph here. Looking at the top, you can see these beautiful hair bundles on the top of the outer hair cells and nice bundles on the top of the inner hair cells. So that's what a normal-looking mouse looks like. But in a mouse that's missing both of the TMHS or LHFPL5 genes, then you can see how there's a missing bundle. This one's looking pretty scraggly. They're really not developing well. They don't look good. And eventually those hair cells die. So we can see that this gene defect in one of these critical proteins that's part of the sound sensing apparatus causes the hair cells not to work. So what we thought is, well, let's just try putting a normal copy of that gene back into the inner ear. And the way that we do that is to inject a little bit of virus into an opening of the inner ear. So in this picture, we're kind of sticking it through this little thing called the round window membrane. And the virus gets into this space here and kind of diffuses around and gets into the hair cells. And what we hope is that the hair cells will take up the virus and get the proper gene inside them. More specifically, the way that we do this is, here's a bit of DNA, and it has not just the gene for this protein, but also genes that turn on the protein, that make the cells want to make it. We take that bit of DNA, as I said, and put it into the AAV, and then we inject the AAV into a newborn mouse right into the inner ear. So how do we tell whether that's doing any good? And there are a couple of ways. One is we can wait 5 days. The cells would have time to make the new protein, and we can see whether we can recover the function in the inner ear. So we can just take the inner ear out. This is a little bit of cochlea that we take out and put in a dish. And we want to test the function of those hair cells and see if they're now working properly. So we have a very simple test where if the hair cells are working properly, they become fluorescent. Here is a mouse that's only missing one copy of the gene, and you can see each of these hair cells, each of these little circles is brightly fluorescent, and that tells us the hair cells are working just great. Here's a mouse that's missing both copies of the LHFPL5 gene, and there's none of them are fluorescent. They're just not working at all. And here's a mouse that's missing both copies, but we've put a good copy back in with the AAV. You can see that not all, but many of the cells are becoming fluorescent, and that tells us that they're starting to work again as a result of the DNA that we delivered through the virus. There are other ways of testing hearing, some that are more functional, and that is to simply play a sound to the animal and see whether that sends a signal to the brain. So you can play a little kind of a click. You might play clicks that are louder and louder and louder, and then put a little wire on the back of the head, and if the ear is working properly and the signal is going to the brain, then we can pick up a very tiny voltage, a little bit of nerve signal, and then amplify it up so that we can measure it. So here is a test like that, and this is time, and here we play a very, very quiet click, and there's no response to the brain. Clicks a little louder, a little louder, a little louder. And so you can see as it starts to get moderately loud, there's a little wiggle, and that is the electrical signal that came from the ear that's going to the brain. And as the click gets louder and louder and louder, we can see this big electrical signal that tells us the mouse is hearing the sound. Here's the mouse that is lacking both copies of that LHFPL5 gene. Quiet sound produces no electrical response. Even a very loud sound produces no electrical response. So that mouse is deaf. But if we take a mouse missing both copies of the gene and add back the gene with this AAV, It's still not responding very well for quiet sounds, but as we make the sound louder and louder, now we're getting a response again. So we haven't completely cured the deafness, but the mouse can hear sounds, at least if they're loud enough. Now, I mentioned that human patients who are missing this gene have no hearing at birth, but they also have no sense of balance. And we wondered whether we would also be able to restore at least some sense of balance in these mice by putting the proper gene back into the inner ear. And I mentioned before that when we put the virus into the cochlea, it gets into this space and gets into the hair cells. But the same virus can diffuse over into the balance part of the inner ear. And we've shown that we can get the virus to go into the hair cells in the balance part of the ear just as well. So how do you test balance? Well, a simple way is to let a mouse swim. And mice don't really like to swim. They don't like water, but they actually swim pretty well, and they'll swim back and forth. And they're using their sense of balance to keep their head above water to be able to get air. And he'll swim back and forth, and he'll go back and forth, and you see that his head is always above water. Just fine. He'll do this all day long, keeping his head above water because his sense of balance is perfectly fine. Here's a mouse missing both copies of that LHFPL5 gene. It doesn't know which way is up. It doesn't know where to put its head. Can't get air. And so we have to rescue it very quickly. And now here's the mouse that's lacking both genes, but we put a good copy in with the AAV, and it's not swimming that great, but it's keeping its head above water. And so that tells us that we've restored at least part of the balance function in this mouse. Now these studies are in mice, and they're preliminary in a way, but on the other hand, there's a lot of activity in trying to treat deafness in mouse models for human kinds of deafness. And you can see one of the first was only 6 years ago, but since then there have been 8 or 10 different studies, many of them here, in which at least partial restoration of hearing has been possible using this adeno-associated virus. So that's all in mice, but we're encouraged that these studies can be taken eventually to human, in part because there's been a very successful gene therapy for blindness that less than a year ago was approved by the Food and Drug Administration. This is a blindness that's caused by a mutation in this gene called RPE65, and investigators at the University of Pennsylvania and Spark Therapeutics use a similar strategy with AAV to carry a good copy of the gene into the eye and to restore restore vision in patients that would otherwise be blind because they have a bad copy of the RPE65. So what are the prospects for gene therapy for Norrie disease? It's not going to be quick. It's not going to be easy. We have to get this to work in a mouse model. We have to test it really well. We have to make sure it's safe. Before anything goes into human, there's years and years of study that the FDA requires before it will approve it. But I think, especially based on work in the last few years, that this will happen and that eventually there will be a gene therapy for Norrie disease. So with that, thank you, and I will take any questions. A question here?
Thank you for what you have demonstrated today. My first question.
Just yell.
Yeah. My first question is the following. The therapy is only for babies or also for adults?
That's a very good question. And you saw, for instance, in the mouse that was lacking the Norrie gene, it was okay at 3 months of age. It wasn't great, but by 15 months of age, a lot of the cochlea had degenerated. So the general conclusion is that the earlier you can get a corrective gene into the ear or the eye, the better chance you have of preventing the degeneration.
Second one is
if I can just say one more thing, though, the encouraging thing to me, in a sense, is that the hearing loss comes on fairly slowly. You know, it takes 10 or even 20 years before the hearing loss is really significant. And so there's a pretty big window of time to try the gene therapy, at least for the hearing loss. Some other deafnesses, the patients are completely deaf by birth, and that's a much less optimistic prognosis.
Where can we follow the progress of all these studies?
Unfortunately, nobody is working on gene therapy for Norrie disease right now that I know of. But there are many people working on gene therapies for other kinds of hereditary deafness. And papers are being published in the scientific journals, you know, every month. We submitted one yesterday on another kind of deafness. So I think just following the scientific literature, and you know, there are very skilled people among your group that can find these papers and then get them out to interested people. 3 or 4 questions back there. You first.
Yeah, I have a couple of questions. One being, functionally speaking, for a therapy like this, would the fact that a mutation was random as opposed to inherited from a mother matter, or would functionally this work the same way?
It wouldn't matter as long as the molecular diagnostics have been done to be sure that the problem is in the Norrie gene. For this particular thing, so I mentioned some of the strategies where you have to know exactly what's wrong and then correct that one piece of DNA. Here it's a little simpler. You just put in the whole DNA for the whole Norrie protein, and that should correct any of the kinds of defects in that gene.
My second question was, because I know there's been success with LCA in terms of gene therapy with vision, restoring vision to, I believe, an adult patient. So how
Or at
is
least
that,
teenagers, yeah.
or at least teenagers.
Yeah.
How is that just because with LCA there is a more gradual loss of sight as opposed to from birth, and they've caught it in the window of time that much damage hasn't been done?
Yeah, I think that's exactly right. That the blindness in LCA, this is the Leber congenital amaurosis that is caused by a defect in the RPE65 gene. That blindness, again, it's a little bit, the cells that are affected there are also kind of creating energy for the cells that actually receive the light. And as they slowly degenerate, then the cells that sense the light degenerate also. So it's a slow progression, and there's time to catch it. Yeah. There was another question here.
Hi, a couple of questions, and I know that they are not easy questions, but if you could answer to the best of your knowledge. So, first one is, so what is the average time that you typically have between trials in mice to trials or to approval by FDA?
My understanding is for the LCA treatment and for the human blindness, that took 20 years between the development and the FDA approval. On the other hand, the FDA, the gene therapy technology is working much better. The FDA is more comfortable with the safety of these. And so I don't think the next one's going to take 20 years. It might take 10, maybe shorter. But it'll be a little bit faster than the first ones.
The
Yeah.
other question is, so you mentioned that to your knowledge there is nobody doing research on Norrie disease gene therapy right now.
To my knowledge, yeah, at least not in the year, but I could be wrong.
My question would be, how can we influence that? So what determines whether there is fine, and I understand there will be the private sector that, you know, they take whatever consideration, but from a university perspective, for example, many of the cases that you were referring to were universities. What determines what first goes?
Yeah. So I would say 2 things. One is you've got to get the scientists interested. They have to see both the real cost to patients and the opportunity, and come to a meeting, get them to come to a meeting like this so they recognize that this is not only important, but maybe something that's they can do. And the second thing is to get the NIH to put money into this. So writing to NIH institutes like the National Institute for Deafness and Communication Disorders or the National Eye Institute and sort of saying, what are you doing in this area? It's not necessarily going to get them to put a lot of money in, but they're going to be more aware of it if they're getting letters from the patient groups.
Does funding help? I mean, is the question of funding, or is more about interest?
It's both. So the scientists have to become interested in the problem, and they have to be able to get money to study it. Yeah. You have a question?
It's more of a comment than a question. And I just wanted to, there is some early research going on in Great Ormond Street Hospital. It's specifically, though it's still very much in the early stages, it is, the hope is that that research is going to hopefully contribute to work in the future. So there is something going on. It's in Great Ormond Street, and I think we're posting updates on the Facebook group about that, but that's sort of one thing to.
Well, that's really good news. She's a wonderful geneticist, and
So much of, and another comment related to your third, very much the interest that she has taken by the Nori community and how people, like Wendy and others who've just persistently been asking and been pushing and, support groups like this, family groups like this, it just always has so much more of an influence on them.
But it is pre-genetic therapy research, or
So it's in preparation for gene therapy research, so it's a PhD program, it's still very much in the first few months, and looking at first the natural history and then what happens in in Norrie mice, and then hopefully towards the end of the PhD start to try some of the gene therapy approaches that have been, I'll be talking about that a little bit, and I can add to that. Ocular genetics side of things, and I don't know to what extent they have started or what stage you're in, but there are people who are interested, definitely,
that's great news. Other questions? There are a lot. Here?
Right. So just to put things very clear, you actually are saying that these gene therapy have a potential to stop the hearing loss from getting worse, but not to reverse it. Is that correct?
I think that's probably true. It's possible that it could reverse it to some extent, but I think the main hope is it would stop the further progression.
Because 3 years ago on this conference, there was another specialist called Albert Edge, who explained by a similar way of injecting viruses in the inner ear as you have been describing, that he could reverse the hearing loss caused in deafened mouse by white noise. So there were not mouse that had any genetic disability or genetic lacking, let's say, but he deafened them using white noise, and then he injected a certain protein or like a signaling path for a certain protein in or a certain DNA, and then the hearing was partially restored. So this is also something that might be very interesting to
That's true, and Dr. Edge and others have made progress in allowing the hair cells to regenerate. So I mentioned that when the hair cells aren't getting the right energy from the lateral wall, then they start to degenerate. So if you can focus on getting the inner ear to grow more hair cells, and frankly that's a much harder problem, then you could possibly restore the hearing loss.
And then I have one more slightly off-topic comment, but maybe because you have kind of pinned down the cause of the Norrie hearing loss here, there is a tradition, at least in Europe, that sudden hearing loss episodes without known cause is treated with hyperbaric oxygen. And I was wondering if providing these, these hair cells that don't have enough capillaries with more oxygen, or at least, yeah, more oxygen than normal, would slow down the reverse of it, if it has any potential for doing any good for that.
So that's an interesting idea, and I don't really know the answer. I don't know whether, somebody breathing a higher percentage of oxygen all the time would be able to maintain their hair cells.
So that was my little hypothesis.
Yeah, no, it's a good idea, and to be honest, I don't know the answer to whether that would work. Somebody should try it. Yeah. Many other questions. Second row, yeah.
So I wonder if you could explain why and don't get the hearing loss. Does their profile still look the same? Are you guessing, and why do, and there's such a range in ages of the men, and so I'm just curious, does the ones who don't lose their hearing age, or it's a later age, does their picture look different, do you think?
Well, we see this with a lot of genetic diseases where I mentioned that the DNA that is the instructions for the Nori protein has 400 different bases, roughly. And it could be that if you make a mutation in this one, it really messes up the function of the protein, and the hearing loss might come on very quickly. You make a change in this one, it actually doesn't matter at all. The protein works just fine. And you make a change in this one, and it messes up the protein a little bit, but it still sort of functions. So each of the different changes can have either very severe or very mild, or in some cases, no effect at all. And so one of the things with any of these hereditary disorders is trying to match the individual DNA change with how severe the disease will be. There's a question here first.
But just to be clear,
Yeah.
though, and it goes back to what he was saying, 3 years ago, one of the lecturers was talking about varicosities in the ear, whereas actually what you're talking about with the capillaries is more arterial inflow. Am I correct?
That's what we saw.
Rather than stagnation on the other side of the capillary.
So we haven't looked at this in a long time. It was almost 20 years ago that we did the work. But nothing new has at least been published on this. So it's still our feeling that the capillaries aren't getting formed properly.
Arterial inflow?
Yeah, but I hope that there's new work that hasn't been published. And yeah.
Do you know why there is variation with the age of the hearing loss, even within the same family? You would think, like my uncle, for example, lost his hearing a lot later than I did. You would think it would be the same variation with the same parts of the DNA that are not functioning correctly. Yeah,
So you would think that, and we don't know exactly why there's variation, but in addition to the Norrie gene, you have about 20,000 other genes. And so there could be differences in what a geneticist would call the genetic background, where in one case, another gene might make up for the defective gene a little bit, and in one case, that compensation isn't really there because of a difference in the genetics. So even with the same gene, we see differences in the severity or onset, and don't really understand it in most cases. Another question.
What's the level of funding that would stimulate further research into the Norrie?
Well, a typical, kind of a standard research grant that would come from the National Institutes of Health brings about $250,000 a year to a lab for 5 years. And it's just very expensive. But getting a couple of those out to people who are working on this would really enhance the research. So that, again, it would be, the big money is at NIH, and encouraging them to support that would be most helpful. Question back there, yeah?
So as far as so for my son, it was obvious that
Yeah. So I think we don't know. I personally don't know the answer because I don't see Norrie patients. But there's a big difference between the eye and the ear in one sense, which is you can look right into an eye and with an ophthalmoscope and see what the blood vessels look like. And there's no way to look into the ear to see what the blood vessels look like. So there's really no way to do that correlation. Oh, yeah. So in the mice where we have put a virus in shortly after birth and then tested a month, 3 months, 6 months later, the preservation of hearing is prolonged. We don't think that that gene is going to go away, although it's not always permanent. But the AAVs are pretty good at just staying inside a cell and continuing to work. Question here, yeah.
So Dr. Sims, she's done so much for Norrie disease patient and our children.
Yeah.
And, future of our research. Who's going to take over that role here? We're a small, and that's where a lot of rare diseases don't get studied because we're a small group. But among families, we have a woman who was on the board who's and she had like 13 blind people in her family or whatever. So, when you talk about rare diseases, as a collective group, and we're getting more and more people all the time because of the Facebook group across the world. So there are so many unidentified people out there with Norrie's children, Norrie adults. But among us, I have a blind brother, I have a blind son. There's people who have multiple people in the families that, so among our group, it's pretty significant. So NIH, I know, looks at the masses, it's like, the football team at the university, they get the money, but then maybe the hockey team doesn't, or whatever. Same concept, it's all about money, kind of. So, how do we, I know you said that before, like, how do we really impart the significance? Because Norrie disease is not only vision and hearing, it's, vascular and other parts of the body that we haven't even touched on. It's really, really serious. And cognition all the way to very, very severe autism. So I work in a vision clinic, and so I know there's a lot of research has gone into not to compare diseases because rare diseases are, they're all important, but there's so many people, more people with RP than there is with Norrie. Yet they're getting lots of research dollars, and they come up with different, types. It's we're really ahead. So.
Yeah, so how do you do that? So, in a way, there's two different questions. How can you get more money into the research, and how can you get a good clinician to focus on this disease and really become an expert in it? Yeah.
And we want a person here.
Yeah. Sounds like you have an answer back there.
Is there merit, though, to making connections between what we know about Norrie and other more common eye conditions, ear conditions? Researcher that there are similarities in some way that other things to which vasculature is common, that perhaps studying Norrie could help in these more common things? Like, is there merit to that?
I think there is, and the NIH, they do pay attention to how large the patient population is, but they're also interested in basic science, and they recognize that advances in gene therapy for Norrie disease could be applied to other hereditary deafnesses or blindnesses as well. So now the drug companies are really counting the number of patients, but the NIH is not so focused on the total patient population. As far as getting, a good physician, that's, I think, just kind of identifying somebody who shows a little bit of interest and encouraging them to take that on as a specialty is the way to do it. You know, you can get a nice ophthalmology resident, for instance, who's kind of just out of training and looking around for something to specialize in, and if you can get that person to say this is, you know, their life's work, that'd be great. Okay? Great. Thank you.
I'd like to thank Dr. Corey for coming out and speaking. I think are down to us, right? How do we organize this? How do we do this? Who do we talk to? It's meetings like this. It's the work that the NDF is doing in the UK, and it's us connecting people and trying to do this, right? We have Florian here trying to help and take over for Kathy Sims to step into those shoes and do this stuff. So, but our part has to be pushed, right? Each and every one of us has to kind of come together and help out. Join us on the board. Come on, take those couple of calls. Find your doctor, talk to them, see what they know, push it, see who they know, right? And if we all work together, we can kind of help do that, whether it's fundraising, whether it's connecting all these people. But that's it. It's on us. So, and I think now we're going to get together and hopefully take a picture with our T-shirts. There's some family greet time, and then I think we'll be breaking for lunch. So